let's take a look how those taste tones actually was transduced into electric signal. As we mentioned, these three modalities, sweet, bitter, and umami, they are GPCRs. 它们是7次跨膜的蛋白 ，and 它们是7次跨膜蛋白的话，那they need couple to the G protein, okay? Couple to the G protein, and the G protein will couple to the another enzyme called PLC beta 2. PLC is phospholipase C. This is different, okay? Phospholipase C is very different from the adenine cyclase or guanine cyclase in the visual system or olfactory system. This one will hydrolyze the PIP2, the lipid, in the membrane, and produce the IP3 and or DAG. This is kind of a second messenger. And then IP3 will open the IP3 receptors in the ER membrane. Then the calcium will come out from the ER. And this calcium, okay, the channel for this taste receptor cells is the triple M5. It's TRP channel, M5, okay? 这是一个阳离子通道。And then the sodium will come in and depolarize the cell. This is kind of for the transduction of the signal. 那其实这个地方this step is not really finalized yet, the understanding, okay? Still quite kind of make some kind of links. How from this PLC beta 2 lead to the activation of this channel. The process still a lot of study focus on this research, okay? And actually this kind of general thing in the field is from the TRP channel the gating. TRP channel is transient receptor potential. This channel actually first is cloned from the Drosophila in the eye. It's because there are some mutations, and then the receptor potential actually was changed, the light detection. And then from there, later people found so important of these TRP channels essentially participate in every aspect of our sensations, like the temperature, mechanical, and also vision, and the taste, and other. Actually, in the brain, we also have a lot of TRP channels, okay, in the brain. They are quite important, okay. So this field actually, the problem is actually how these channels can be gated by this cascade. People still don't know the answer. There may be about a few decades of the study already developed for this research, but the answer is not clear yet. Okay, people claim maybe some kind of lipid directly inhibit the channel, but then if you hydrolyze the lipid, then the channel open. And also there's some kind of this kind of second messenger pathway, and some actually proposed actually even in the visual system in the fly, maybe some kind of mechanical sensation from this TRP channel. Okay, so for the sour and salty taste, then it's more straightforward. Just ion channel there. Okay, they have the ion channel in the membrane, and then the acid can directly go into the cell, depolarize the cell. That's it. Okay. The taste receptor cells, we talk about it, they are not neurons. And they transduce the signals, the taste signals. And then they need relay that signal or transmit the signal to the brain, right? how can transmit to the brain? This is quite complicated, okay? This system is, I guess, actually is why the taste actually, the understanding is so limited. And this actually is quite one of the main reasons actually make the system actually not so easy to study. For example, the taste receptor cells in the taste bud, one taste bud can up to about 100 taste receptors, okay? And then those taste receptors can, based on the morphology, and they can be categorized as type three types of receptor cells, okay? Of course, there are another one type is actually not matured one, because as we mentioned, these cells need to be regenerated, right? And some cells may be at a very early stage, not mature, and then that called type 4, okay? But for these types, these three types, they are matured neuron receptor cells. For this one, this type 1 receptor cells, and then they're quite special, okay? What's special for them? They have the GLUT. This is for glutamate transporter. And also they have the NTP DACE2. This is for the transport of the ATP. And then also they have like a NET for the neurotransmitter NT uptake. So as we know, actually in the brain, when you have this lot of neurotransmitter transporter, it then most likely they're glial cell, right? 就是像那些glial cell is 中文是什么来着？胶质细胞 right. 那so for this type 1 cell, most likely they are the 胶质细胞，像我们 brain 里面的 ，OK？ 但是呢 ，but they actually may be responsible also for the salty taste. Okay, quite strange already. Okay, take a look about type 2 cell. This is the type 2 cell. Type 2 cell, they can express the G protein and also the triple M5. Triple M5 is what kind of channel? It's responsible for the detection of the sweet umami sugar, right? So this type 2 cell then is responsible for this detection 甜的、鲜的、苦的， and they have this kind of signaling molecule expressed within the cell, like receptors, right? They also have the G protein, have PLC beta 2 and triple M5. And this is the type 3 cell. Type 3 cell is quite interesting. Within the taste bud, only this cell can have a direct synaptic contact with the axon from the ganglion cells. So there is a gustatory ganglion cell send the axon to the taste bud, right? And only these type 3 cells can really have a synapse with them. The synapse actually is the EM structure. You can take a look. It's presynaptic vesicle and postsynaptic region, right? And then this is the only type of synapse can be formed between the receptor cells and the gustatory ganglion process.